Good afternoon, everybody. I'm Vertes from the George Washington University. It's wonderful to be able to virtually share my thoughts and this presentation with you. With proteomics, spatial biology has a new challenge. With proteins, there is no amplification mechanism, so it means that we will face a sensitivity challenge. However, there are several ways to overcome this. There are two major um, methods, approaches currently available. Of course, single cell proteomics is available for isolated cells. There are really wonderful studies showing more than 1,000 proteins identified in individual human cells. However, when it comes to tissue-embedded cells, this requires different techniques. So in this case, the optical approach is really with a fluorophore with a cleavable linker, and then you go through this several cycles to tag cells with antibodies and the corresponding fluorophores, and then do the imaging and then cleave the linker and move on. So this enables the coverage for hundreds of proteins. Another approach is a laser ablation-based approach where mass cytometry is employed. Rare earth reporters, rare earth metals are used to tag individual cells where the antibodies decorating these decorating these cells attach these rare earth reporters to those cells, and then the cell of interest is ablated, and the ablation product is entrained into an ICP-MS, and the rare earth element is identified. So this has a very interesting application, for example, in this breast cancer tissue section showing tissue heterogeneity. Now, spatial biology at the metabolomics level really dates back several decades, although not with single cell resolution. So in the early 1980s, SIMS, secondary ion mass spectrometry, was already capable of subcellular spatial resolution and enabled the mapping of small metabolites and elements within individual cells. However, this always brought about extensive fragmentation. So the identification of metabolites was really shaky. It was not reliable. More recently, there have been improvements with cluster ion sources, but that's not where we are going with this. In the 90s, MALDI, matrix-assisted laser desorption ionization, became available, and this was very useful in terms of not fragmenting those molecules, those metabolites. The spatial resolution is somewhat lower, only 10 to 15 micrometers typically. And also these imaging approaches were practiced on a rectangular grid. So over the tissue, people were analyzing the surface point by point, and they were collecting spectra for every point, and that became a pixel in the mass spec image. The metabolite with a particular M over Z, its intensity was mapped on the surface, and that was the basis of forming the image. Of course, what is lost in this is the rendering of those intensities to individual cells. Also, there are some technical issues here that means, needing to put this tissue in vacuum, which is obviously not conducive to live specimens. Further advances were were shown when ambient ion sources came into play. This was in the 2000s. Various ambient ion sources were introduced here. There is an example from my lab, laser ablation electrospray ionization, or LASI. And we were able to image, for example, in this case, a coronal brain section from a rat, and identified multiple metabolites and lipids in that tissue section. The good news is that at this point we were not stuck with having to put our sample in vacuum. So we were operating at ambient environment. Also, we were using fresh or frozen tissues. So this was closer to reality. However, it was still reliant on a rectangular sampling grid. At that point, we were interested in really targeting individual cells. And we constructed this microscope that combined the lazy sampling and analysis with an optical microscope. In order to be able to target individual cells, we used a sharpened optical fiber that was pointed at individual cells for ablation. And as over here on the right, you can see how these individual cells burst open, and the analysis was conducted on the ejected cell content. You can actually see here, as we are moving from cell to cell, this is the tip of the optical fiber that delivers the laser pulse, and ablation takes place after a few laser pulses. So in this case, we were able to get an analysis from 1,084 epidermal cells from onion. On the right, you see here a typical spectrum. There are many more peaks in here, just they're just not shown because of the intensity of these disaccharide and other strong peaks. For example, you can follow a selected ion over time. In this case, the malate ion was monitored throughout the entire analysis. So when you collect the intensities for let's say 1,000 cells, you can of course plot the intensity distribution for the entire population. And we did this, and we've observed two major features. One was really the shape of the distribution itself. Many times it was log normal, like in this case, the glutamate and the allele intensities showed a log normal distribution. Other times it was Gaussian. But then there was some cases when we observed a bimodal distribution, which was very interesting for us. In this case, the disaccharide ions show the bimodal distribution. To characterize the unimodal distributions, we have introduced this concept of metabolic noise, which is really characterizing the width of these distributions in relation to the mean value. So you can see the definition over here. If you look at, if you assume that this metabolic noise has something to do with the actual concentration of a particular metabolite in the cells, you can actually test that hypothesis, and that's what we did here. So you see the molarity of various metabolites in the cell and the metabolic noise on the log scale. And you can see that really there is very little, if any, dependence on the concentration of metabolites. Another question we were wondering about was, especially in the case of this bimodal distribution, is there a relationship with the cell volume? So is it possible that larger cells that are um, more mature, or maybe in other cases closer to division in the cell cycle, is it possible they exhibit a different metabolite composition? So we measured using morphometry, we measured the cell volume distribution that turned out to be also log normal. It's shown here. And then we were able, because we could render the analysis of the cells to their cell volume, to the cell volumes, we were able to separate out from this bimodal distribution small cells with small volumes, medium volumes, and large volumes. And as you can see, we observed that the small cells um, did not show a bimodal distribution. They exhibited a unimodal distribution with the higher mean value. Obviously, for the bimodal distribution, you have one mode that has a higher mean value for the metabolite concentration, and then there is another one with a lower one. So the small volume cells exhibited the unimodal distribution. Medium volume cells had both modes, whereas for high volume cells, the situation was unclear because we didn't really have enough statistics. Only 58 cells fell into this category. The next question we wanted to explore was, how many cells were needed really to actually have reliable statistics to determine these distributions. In this example, we took the fumarate data set, all 1,000 cells, and we knew that this actually exhibited a bimodal distribution. It's shown down here. And then we artificially and randomly removed downsampled distributions, the cells to 600, 400, 200, 150 cells, and try to construct the corresponding histograms. Obviously, we were interested in our ability to discern this bimodal nature from these other downsample distributions. And as you can see, you can visually assess this and come to the conclusion that somewhere around 200 cells, below 200 cells, you really lose the ability to discern the bimodal distribution. Now, of course, the number of cells you need will also depend on what kind of distribution you're trying to discern. If the underlying distribution is unimodal, you probably can get away with fewer cells. So since we were able to identify, we had a metric for cellular heterogeneity for metabolites, which is the metabolic noise, as I've introduced it in a previous slide. We wanted to see how different metabolites fared with respect to this metabolic noise. So you have a list here. We assessed this for a bunch of metabolites. And as you can see, there are some here that have decidedly high noise when the metabolic noise as defined here is larger than 0.5. We call that high noise. And then we have some cases when metabolic noise is relatively low, less than 0.5. One can argue or wonder about the origin of these differences between the metabolic noise values. One hypothesis is that low and high metabolic noise may have to do with the tightness of the regulation of that particular metabolite in the metabolic pathway network. And then we took a closer look again at the bimodal distributions and noticed that when you decompose it into two subpopulations, we have this high mean and low mean values. And this is really an indication of two subpopulations, two uh, distinct subpopulations, although they are hidden. When you do bulk analysis, they are hidden. You wouldn't be able to discern these two subpopulations. So we call these hidden phenotypes or hidden subpopulations. Um, Another example we were exploring was the root nodule cells in the soybean plant. This is particularly interesting because it's a very heterogeneous tissue. You have intermingled infected cells infected by rhizobia, soil bacteria, and some cells that are not infected by rhizobia. As you can see here, we are ablating individual cells again and collected the corresponding spectra and analyzed them separately for infected and uninfected cells. So if you do hierarchical clustering, very quickly you realize that uninfected and infected cells, as well as, excuse me, epidermal cells can be distinguished. When you construct the histograms, it's also clear that again you have cases when you have unimodal distributions. In this case, both normal and log normal distributions were observed, but then in other cases we observed bimodal distributions. We were wondering, what can be a reason behind these bimodal distributions in these infected cells. And we came up with a hypothesis for the case of NAD+. If you go to the literature, you can find that NAD+ to NADH ratio is different in quiescent cells and in dividing cells. So we argued that in these root nodule plant cells, the bacteria, which are measured together with the plant cell, the individual plant cell, either are still in the dividing mode when they haven't filled the entire plant cell, or when they do fill the plant cell, they go into the quiescent mode, G0, because they have no more opportunity to divide because of the geometrical constraint. Finally, we also were interested in image segmentation for rare cell analysis. So this is, again, a root nodule with the infected cells identified. Through image segmentation, we were able to do morphometric analysis and pick out cells based on their size. So we selected 25 cells that were on the larger side, and we were curious if we were able to selectively target those cells and analyze their composition. Indeed, that's what we did. As you can see here, automated analysis was performed of 25 selected cells, and we were able to map the metabolite intensities for these different metabolites back onto the image. So in conclusion, we showed that optically guided mass spectrometry imaging can be a useful tool for spatial metabolomics. We showed that this fiber-based lazy mass spectrometry was capable of the analysis of single tissue-embedded cells for metabolites. We found metabolic noise, which is a metric for cellular heterogeneity for unimodal distributions, for metabolites with unimodal distributions, whereas for bimodal distributions, we found latent subpopulations, and which you can also correlate with hidden phenotypes or metabolic states. Finally, I want to thank my group who made this work possible, as well as the funding agencies, DOE and National Science Foundation in the US that were funding this work, and our collaborators at the Pacific Northwest National Lab and the University of Missouri. And finally, thank you for your attention.